Hi, this is Dr. Ricardo of the Department of Anatomy and Cell Biology, University of Cape Coast, and I present to you Embryo-genesis week 1, 2, and 3. So if the egg is lucky and there's fertilization, secondary oocyte, which was arrested in metaphase, will complete the second meiotic division and a zygote will be formed. After that, a two-cell blastula will also be formed, and then four-cell blastula, then a morula, then some species appear within the morula that will cause the blastomeres to move to the North Pole, what I call the North Pole. And then you have the space appearing here. So that will become the blastocyst cavity. And then all the blastomeres will be pushed towards the North Pole, forming the inner cell mass or embryoblast. So that is what occurs. And then at the end of the day, the blastocyst, as this is called, because it now has a blastocyst cavity and inner cell mass, and then some of the cells that are surrounding it, it will get to the endometrium by day 7 and start implanting. At that point, you end up having this spherical, nature, having finger-like projections. The spherical nature with the single layer, that is the trophoblast. But once the blastocyst together with its trophoblast gets, contact with the endometrium, it starts differentiating into syncytial trophoblast. And then the rest of the trophoblast will become cytotrophoblast. So that is what we see here. That is syncytial trophoblast and then cytotrophoblast. The difference between the syncytial and cyto is that the syncytial is multinucleated, which means that it has several nuclei, okay, which have not been partitioned. It has one cell membrane. Unlike the cytotrophoblast, that will have individual cells with their own, cell membranes. So that is what it is. As you can see, you can count several cells here. So each of them has its own cell membrane. So this is a scanning electron micrograph showing how the blastomeres appear, okay, like balls. Okay, so that is how you see it. So again, that is a 2-cell stage, 4-cell stage, 8-cell stage, morula, with the blastomeres there, and then you have the inner cell mass, blastocyst cavity, and then that is the inner cell mass. I'm trying to get a pointer to help with the pointing. So let's see how it goes. All right, so you now have the you have this 1 to 2-cell stage, 4-cell stage, on and on, but this is just colored, okay? So it's a bit fancy. So that is how this will look like with the inner cell mass. It's actually 3D. Yeah, so again, you can see that's 2-cell stage. And then you can even see the other additions, okay, including the extra blastula that are adding up. So 4-cell stage, 5-cell stage, on and on and on it goes. And then you have the polar body. That's what you can actually see here in a purple color. So for the cytotrophoblast, which now has its individual cells, and then the syncytial trophoblast coming out, both of them coming out from the cytotrophoblast, because the blastocyst has an encounter with the endometrium. All right, so that is what is ongoing, and the deep green is the syncytial trophoblast, which is actually as a result of the trophoblastic cells differentiating, okay, and forming finger-like projections, and they are multinucleated because there are a lot of nuclei within just one cytoplasm and cell membrane, unlike the cytotrophoblasts, which are actually uninucleated. So the functions and properties of the cytotrophoblast and syncytial trophoblast. The syncytial anchors the conceptus, okay, to the endometrial wall. The cytotrophoblast is a source of nutrition for the developing blastocyst, okay? So tropho means to feed, okay, or trough means to feed. So you can see here that the inner cell mass, which is blue, is now having an additional layer, yellow, there, known as the hypoblast. So we are actually entering week 2, where you have 2 structures being formed. So you have the embryoblast there, now you have hypoblast, which is also termed the primary endoderm. So embryoblast is inner cell mass, forms epiblast and hypoblast. The 2 together will form the bilaminar disc. So this is just a summary of what we have seen so far, and you have it here. Fertilization, and then you have your morula, embryoblast, and then the hypoblast also differentiating, and the blastocyst cavity of blastocele as a trophoblast, which will eventually become the cytotrophoblast. Then you now have ectoderm and endoderm, as we will see. It's just a summary, so we are one step ahead with this diagram as the amniotic cavity, which will end up forming the 3 germ layers, ectoderm, mesoderm, and endoderm, called the embryonic disc. You have the amniotic cavity with the amnion, and then the yolk sac also there. So yes, this is what happens. Inner cell mass, blastocyst cavity. Since the trophoblast has started its formation, you have inner cell mass, the hypoblast is at the bottom, then you have cytotrophoblast. So if you take this section, which is towards the head region, the cephalic region of the embryo, you have the epiblast and hypoblast like that. And you can see that the cells are columnar in shape. Okay. And then the portion of the hypoblast, which is also columnar in shape, will form the prochordal plate. The future mat will be there. And then the rest of the hypoblast that are cuboidal in nature, together with the epiblast, will form the bilaminar disc. So syncytial trophoblast produces hormone, the human chorionic gonadotropin. HCG maintains the hormonal activity of the corpus luteum to maintain pregnancy. Enough HCG is produced by the syncytial trophoblast at the end of the 2nd week to give a positive pregnancy test, which usually is positive when you put urine on it. Okay, this serves as the basis for pregnancy test, so it can be excreted in urine, and then it can give a positive test. exocoelomic membrane converts the blastocyst cavity to an exocoelomic cavity. So yes, this is what we're talking about. So you can see that there's a yellow, lining within the blastocyst cavity. It connects the hypoblast. Because of the presence of that yellow lining, which is also called the Hughes membrane or exocoelomic membrane, the hitherto blastocyst cavity changes its name to exocoelomic cavity. So exocoelomic cavity is just like a blastocyst cavity, which has been lined, okay, by a new curtain, if you like. So in your room, you can buy a new curtain to just beautify the place, and you can decide to change the name, okay, from old to new, right? And you can also see that there's a space appearing above the epiblast, which will form the amniotic cavity. All right, so if the yellow partition, or for want of a better word, the yellow layer is laid down in the blastocyst cavity completely, you have your exocoelomic cavity. Your bilaminar disc is there with epiblast and hypoblast, and then now the amniotic cavity has become larger now, as you can see, with its amniotic membrane also, covering it at the roof. So that is what you see here. So more or less, the epiblast is forming the floor of the amniotic cavity, okay, the epiblast. Exocoelomic cavity is now called primitive or primary yolk sac. Remember the yellow lining of the blastocyst cavity converting it to exocoelomic cavity. Now the exocoelomic cavity is going to change its name to primitive yolk sac or primary yolk sac. You can still see the epiblast and hypoblast and amniotic cavity there. You can also see that this time around, the cytotrophoblast has encircled the whole developing embryo, okay, the light green. And also the syncytial trophoblast is almost completely encircling it as well, okay, because the conceptus is eating into the endometrium. So as it keeps moving into it, the surrounding cells envelope it, okay. So eventually, you are going to have the yellow, the light green, which is the syncytial trophoblast, the cytotrophoblast, and then the deep green, which is the syncytial. So let me go over again. Eventually, you are going to have yellow, light green, and deep green. The yellow is the exocoelomic membrane. The light green is the cytotrophoblast, and the deep green is the syncytial trophoblast. Also, see that, or note that there are species called lacunae appearing in the northern portion of the syncytial trophoblast. So this is going to start collecting blood vessels, okay, and then eventually the placenta is going to form. So by day 10 and 11, blastocyst has completely implanted, primitive placenta circulation is established. So as you can see, now you have lacunae network, and blood has started filling those lacunae in the syncytial trophoblast, right? Then you can see the amniotic cavity with the amnion and the epiblast, hypoblast, primary yolk sac now, because we have changed the name from exocoelomic cavity to primary yolk sac. And you can also see that the cytotrophoblast has encircled the entire developing embryo. And then in addition, something has filled the space between the exocoelomic membrane, the yellow one, and then the cytotrophoblast. So in between them, you now have a solid filling, okay? Either to the cytotrophoblast was in contact with the exocoelomic membrane or the yolk sac now, but now there's a solid feature, what we will call as the extraembryonic mesoderm. extra-embryonic mesoderm fills in between the yolk sac and then the cytotrophoblast. Extra-embryonic mesoderm so that is the red portion. You can see that you have amniotic cavity, epiblast, hypoblast, and then you have the definitive yolk sac developing, but ideally it's actually the primary yolk sac, okay, which will later on become secondary yolk sac. So that is what is supposed to be at this point. The deep green is still in situ, the light green is cytotrophoblast. And now you can see that there are some spaces appearing within the extraembryonic mesoderm, and those spaces are going to add up or join to form one big space. Okay, so that space will now be called extraembryonic coelom, and it's going to split the extraembryonic mesoderm into two. One that will be very close to the yolk sac and one that will be close to the cytotrophoblast. Okay. So the one that will be close to the cytotrophoblast will be called the extraembryonic somatic mesoderm or somatopleuric. And then the one that will be closer to the yolk sac will be called the extraembryonic visceral mesoderm or splanchnopleuric. The part that does not split becomes the connecting stalk. Also will form the umbilical cord. And now you can even appreciate that the syncytial trophoblast has encircled the whole developing embryo. You can see that. As for the cytotrophoblast, it has done that already. Chorion, so wall of the chorionic cavity. It consists of the somatopleuric, that is the extra embryonic somatic mesoderm, the part that is closer to the cytotrophoblast. Then followed by cytotrophoblast itself and then syncytial trophoblast. So those three layers will form the chorionic membrane. The hypoblast produces additional cells that migrate along the inside of the exoslomic membrane. These cells proliferate and gradually form a new cavity within the exoslomic cavity known as the secondary yolk sac. So like I said, the primary yolk sac that will be converted into secondary yolk sac. So you can see that the arrow is pointing at the fact that the primary yolk sac is now constricting at a point where it will pinch off portion of it. Okay, the rest will be the secondary yolk sac, and it has been lined by an additional, membrane. Okay, which has been lined, so it is now even thicker than the first exoslomic membrane. Now you have an additional membrane lining it, so it will change from the primary yolk sac to secondary yolk sac, and then it will also pinch off. So portions of the primary yolk sac will actually pinch off and will not be part of the secondary yolk sac. Whatever is left now will join and become the secondary yolk sac. So you can see that the remnant of the primitive yolk sac is actually getting to the bottom, like at 6 o'clock, if it is a clock, you see there, and the rest become the secondary yolk sac. You can also see that maternal blood has now filled the lacunae within the syncytial trophoblast, establishing the placenta. Then you can also see the connecting stalk where there's no splitting of the extraembryonic mesoderm. Become connecting stalk and then umbilical cord eventually. So this is what you see. Now you still have epiblast, hypoblast. You now have the yolk sac, which is secondary yolk sac. Then you have the extraembryonic visceral mesoderm, which will surround the yolk sac, the extraembryonic somatic mesoderm, which will be very close to the cytotrophoblast. And then you have the cytotrophoblast itself and then syncytial trophoblast. So those three will form the chorionic membrane. And then the big space between the chorionic membrane and then the extraembryonic visceral mesoderm, which is close to the secondary yolk sac, becomes the chorionic cavity. So the chorionic cavity is actually the cavity that came out as a result of the spaces that appeared in the extraembryonic membrane, somatic mesoderm, okay, which coalesced or joined to become a bigger space. Now that is the chorionic cavity. Remember, that is what separated the extraembryonic mesoderm into somatopleuric and what's planktopleuric. So in the nutshell, that's what we've done. Stage 1, fertilization, then zygote, then morula, early blastocyst, late blastocyst, inner cell mass. Then on and on you go. You have the bilaminar disc, primary yolk sac. Then eventually you have the splitting of the extraembryonic somatic mesoderm into two to form planktopleuric, and then what? Somatopleuric. And then also the primary yolk sac is lined again by a layer, and it pinches off, and then it forms the secondary yolk sac.